Hello everybody, welcome to topic 7 of our IB biology content. Let's start out with the essential idea like we try to for every new subtopic, and that is the structure of DNA is ideally suited for its function. And as we will see, if we look at a very, very basic diagram of DNA, we'll see it looks kind of like a ladder with rungs in the middle. These outside parts are made out of sugar and phosphate, and the inside part is made out of nitrogen bases. And a lot of this hopefully is review from last year when I believe topic 2 we went over the function of DNA and everything. So remember the structure, which is a double helix. We'll get more into that as we go through. But the function is to store information. And by the way, I'm going to try different colors, and I don't know, I use red a lot, and red's kind of harsh. It's a little, it clashes a little bit, but you can see it. So I might switch between different colors. Last year I did online. Feel free to give me, hints and pointers as to what works best. So the basic structure, we know if we do a more sophisticated diagram, it's going to be a double helix. It's 2 strands twisted, right? And with a nitrogen base in the middle. And it's great at storing information because these nitrogen bases are made out of A's, T's, C's, and G's. And your body uses this to store the information. And remember, the name of the game is taking the DNA, turning it into RNA, and then turning that into protein. That is what we are here for. That's what we need to do to stay alive. So the nature of science, like I try to tell you all the time, not all the time, but pay attention, observe things. That is so crucial for biology and to all of your subjects and just being successful. Pay attention to stuff. Don't be the one on your headphones all the time, zoning out. Be aware of your surroundings. Ask questions. Really look in, like when we do biological drawings, really carefully look at whatever the sample, whatever the specimen is, and really try to get the nuance of everything, because normally we think in patterns and we just kind of go right to those patterns, and we don't see the details we should. And probably the most crucial thing that allowed Watson and Crick to finally, on top of other people's information, discover the structure is Rosalind Franklin's DNA diffractions photograph. She's holding it right here, but I'll show you the next slide. But basically taking x-rays, which you got, I just had a cavity filled here, but that came out because of an x-ray they took a while back, and that's been used for a long time to study the crystal structure of proteins, nucleic acids. And she was the first to observe this very peculiar pattern that the DNA created under the X-ray diffraction device. And you see it created this X, and if you're looking down through the DNA, you will see an X shape as the twists turn in as the DNA twists, and the nitrogen bases, as they're joined in the middle, twist around, it will form an X pattern. And that's what really put it together for Watson and Crick. They were really, really stuck until they came upon Rosalind Franklin's photograph, X-ray diffraction photograph. But they tried to cover up that it wasn't really her idea, because they were just sexist, pure sexism. They did not want to admit that a woman gave them their vital information. They thought that would be detrimental to their own psyche and to their reputation. So that's disgusting. But hopefully, I know that's still happening. A lot of this research is male-driven still to this day, but it is getting better. But from, you see that X-ray, that kind of cross pattern I mentioned before, we could tell, or we could, scientists could tell that it forms a helix based on that. Yeah, you can kind of see these little dashes that indicates a helix. The size, the amount of twists they're able to figure out, how many bases per twist, and the double-stranded, because some people thought it may be 3 strands or still 2 strands. Not 2 strands so much. They knew RNA was single-stranded, I believe, at that point. But also, they didn't quite know where the phosphates were. So this gave them the information the phosphates are on the outside. All very important information to come upon. So the first understanding is that nucleosomes help to supercoil the DNA. And DNA is really long, it needs to be organized, and the nucleosomes help to do this. Man, my handwriting is so much better with my trackpad, or my little graphics pad, than my Surface. I gotta fix that. So this is a nucleosome right here. The DNA wrapped a couple times around a group of 8 histone proteins. There's 8 of them. So there's 4 on top, 4 on the bottom, basically. And that's how DNA is organized. When it's what I like to say, it's like a hose, a garden hose. If you twist it around something, it's organized. If you don't, it just spreads out. Not only is it disorganized, it takes up a lot of space. So this helps so that the DNA takes up less space. So you can maybe have some more mitochondria or some other things that'll help keep the cell alive. Another is the structure helps suggest a mechanism. You have those 2 strands. So the idea is all you got to do is separate the strands, which we do in this step down here. So you take the original DNA, the parental, and you separate it, and then you just bring up free nucleotides and they match up. And you'll see this word template a lot, and I think you're familiar with that, but it's basically a model. So if you have this A exposed, a T base is going to match with it. G is going to come match with the C, and so forth down the line. And the beauty of that, you always get identical molecules. For life to really survive successfully, it needs to replicate itself. So I'm going to put the note, very important for replication. You have these identical molecules. And without replication, you're dead. And I'm not just meaning, not so much, replicating the organism, like reproduction, but just, your skin cells have to be replaced constantly. You get injured at all, your body has to replicate those cells. So yeah, enzymes use each strand as a template. Just like everything else, enzymes, run this process of DNA replication. We'll get into those enzymes later, of course. And when they're identical, we say they complement. Let's see, wow, getting used to my old system from quarantine. Complementary strands. That means just they're identical. Like what it says below here. Complementary. All right. Now, crucially, it only happens, DNA only gets copied in a 5' to 3' direction. Now, it's all based on the sugar, the deoxyribose sugar. This is an oxygen. These numbers refer to the carbons. There's a carbon here, 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 and right here sticking kind of off. So this is the 1 carbon, this is the 2 carbon, this is the 3 carbon, 4 carbon, and 5 carbon. So that's why 5 prime is up here, because this right here, let me draw an arrow, boom, that's the 5 carbon. And down here, this carbon right there is the 3 prime carbon. So this would be, if this were a DNA molecule, it would be produced in this 5 to 3 direction. DNA polymerase can only add nucleotides to this 3 prime, and it can't add to the 5 prime, and I learned once why that was. Structurally, can't remember right now. Sorry. Maybe I'll add something to this later. So 5 to 3 prime. And just if you're curious, when DNA or an RNA is replicated, it also goes in 5 to 3 direction. So thank goodness something is easy to remember, right? So the DNA unzips, separates, and you have basically 2 sides, right? We call it, one side is always the leading strand, another side is always the lagging strand. And the reason is, since remember, it can only go from 5 to 3, this other side, it can't go in this direction, because that would be from 3 to 5. So it has to wait until it opens up and then go backwards kind of. And as it starts to do that, a whole bunch of new DNA opens up, so it keeps having to make new fragments. And you got to remember this name, Okazaki. So try to remember that. That's important. They try to get away from naming, in science, having everything be named after somebody, but not this time. Since this one does not need to go in fragments, it's called the continuous strand. And over here is called the discontinuous. So continuous meaning there's no break in the DNA. It just goes along its merry way. The DNA polymerase just keeps making new DNA. We'll have a big diagram about this later. And down here you have these fragments, because remember, only can go in that one direction. Here we have the fancy diagram, and all these are all you need to know these. So practice these. Practice. And some of them should be somewhat practice. What is that? A lot of these should be somewhat familiar because we went over basic replication last year, but you need to practice again because last year was last year. So the helicase right here breaks hydrogen bonds. The H stands for hydrogen. Between the base pairs in the middle. It's a fairly strong bond, but not as strong as, say, the bonds within a molecule. And the main workhorse for all of this is this DNA polymerase. That's the main enzyme, I would say, that makes it happen. That's the main character, right? I would say like Tom Cruise or Brad Pitt. You guys don't know that. So, oh man, who's like the main? That Captain America guy, Chris. One of the Chris's. He would be like, that would be like the main one. Or what's the Captain Marvel? The Captain Marvel of this whole show. And remember, they attach only in the 5 to 3 prime direction that we saw in the last slide. You have this RNA primase. That you have to have RNA begin the process. So that kind of shows you there must have been RNA before DNA, because you can't even copy DNA without RNA. So that's pretty good evidence for that. Initiation, RNA primers, it gives you that initiation sites for the DNA polymerase. And when we talk about the main polymerase is this DNA polymerase 3. They should have called it 1, but they discovered it 3rd. So they named it, they just named them in the order they discovered. So DNA polymerase 1 removes you gotta get rid of the RNA. You can't have that RNA in there when you go to make your final DNA. So you get rid of it. The DNA polymerase 1 does. And then the DNA ligase, Okazaki, kind of glues all those Okazaki fragments together. It's kind of a big mess of different chunks. Okazaki, or the DNA ligase, I like that name though, comes in there, glues them all together. And here you have the whole thing. So hopefully you tried to label this before here, but this is the topoisomerases. Don't touch your face, everybody. Trying to keep that on trend for me. Isomerase. Now IB they'll probably stick with topoisomerases since it's in this, but it's also gyrase called that. And kind of going in order here. Let's see. Right here, you have the DNA helicase, separating. So the topoisomerases kind of keeps everything organized so it doesn't get twisted. And that makes it easier for that helicase to come and untwist everything. Then you have the primase come in and put that primer. This right here is that RNA primer. And now that you have that RNA primer ready to go, you can see on both strands you need that RNA. Then you can have the DNA polymerase come on in there. Remember, that's DNA polymerase 3. So remember, DNA polymerase 3, that's kind of the main person there. And let's see. We got this other part down here, this also DNA polymerase, and it's kind of confusing because they're two different colors. And then remember, you got to glue together all of those little strands, and that's what that DNA ligase is for. And remember, this is the continuous one here, and it might be on the bottom in some graphs, so don't think it's always going to be on top. Always make sure you look at it. So this is the leading strand up here, and then this is the lagging strand.